Thank you, Jane. Thank you, everybody, for inviting me here today. So, like Jane mentioned, I'm Stefano Cataldi. I'm a PhD student in the neuroscience program at UBC and working now in Dr. Matthew Farrer lab at the Center for Applied Neurogenetics. So at the Center for Applied Neurogenetics, what we do is we gather information from clinical, so from Silky and other clinicians, we get all the information about what Parkinson is, how it presents. We study the genetic of this population because in some cases, Parkinson can be caused by genetic mutations. We can use those mutations, those alterations, to create mouse models that we can study to understand what is happening when the mutation is present. We can look at this in animals and in a single cell, and see what's the mechanism, and use these models to develop treatments and biomarkers to bring back to clinical, so we can use it to identify specific population of risk or to treat a specific population. So starting from clinical, we already heard today about how Parkinson presents and all the symptoms, and you probably know all of this already, but just want to point some specific aspect of Parkinson. We know that at time of diagnosis, Parkinson presents with loss of dopamine. Dopamine is the main neurotransmitter that is lost in Parkinson disease. We also have at this time of diagnosis the typical motor symptoms, tremor, slowness of movement, postural instability. Often we see also mild cognitive impairment, and in later stages we can see dementia. This stage, the dopaminergic neurons that we lose are mainly in an area of the brain called substantia nigra, which in Latin means black matter. And you can tell in the figure there, the brain on the left is a healthy brain, it has a black stripe of dopaminergic neurons, but these are lost in the brain of the right, which comes from a person with Parkinson's disease. At the same stage, we also see the pathology already mentioned, which is often aggregation of the specific protein called alpha-synuclein. So this protein can aggregate in this formation called Lewy body. So when it's not folding properly, it can aggregate, and we see this in brains from people with Parkinson's disease. We also know that there is an earlier stage of Parkinson that often presents with non-motor symptoms. So we have constipation and loss of smell that we already mentioned today. We had mood disorder. We had sometimes cognitive impairment already. What we don't know is what is causing Parkinson's disease. So we know that there are risk factors, there are environmental factors and genetic factors, and probably a combination of all. So we have the presence of these different factors that then lead to the disease. So this is where we can look, we can use genetics to understand this, what's happening in these early stages of the disease. So we can look at people's genome, so a specific sequence that is making who we are. And these specific traits, this specific genetic composition that we have, we pass it on in the next generations. So we can look at this, we can screen the genome of people, and what it looks like is pretty much a sequence of specific what we call nucleotide. Here shown at different letters, G, A, or C. And so we can look at this sequence, and sometimes we see that there is alteration, there is some nucleotide that is changed with another one. So what this is causing is producing a different type of protein. A protein that is functioning in a different way. So for example, the most common genetic mutation linked with Parkinson's disease is a mutation in the protein called LRRK2 up there. And there is this substitution that is changing the conformation of this protein. What's interesting is these proteins are often conserved through evolution. So we can see the same protein in mice, rats, and other animals. So we can use these models to study the same protein because it's doing the same things that it's doing in humans. So obviously, many genes have been linked to Parkinson over the past 20 years. And for this talk, I'm going to focus mainly on alpha-synuclein, which we already mentioned, aggregates in Lewy body in the specific pathology of Parkinson's disease, and mutation in these proteins can be cause of Parkinson's disease as well. So not only is found in non-genetic cases of Parkinson's disease, but can be the cause of the disease itself. LRRK2 is the most common genetic mutation with Parkinson's disease, and BPS35 is the actual main focus of my project. It's been found by my group, a mutation BPS35 in families carrying Parkinson's disease, and we're studying this protein in our models. So now that we have this genetic information, we can actually identify a specific population at risk. We can tell before the disease is present that someone has the same mutation, so most likely will develop Parkinson's disease later on. And when we look at these carriers of the mutation, so this is a study actually from UBC, from Metzger group, we actually see that in carriers of this mutation, dopamine is actually increased in earlier stages. So these people don't have Parkinson's disease yet, most likely will develop Parkinson's disease later on. They have increased dopamine release, the same neurotransmitters that then is lost in the presence of the disease later in the age. So I'm talking about dopamine, I'm talking about neurons. What are neurons? Neurons are these cells that compose our brain, and they have to communicate to each other so we can function properly, so we can have all our behaviors, motor behaviors, and learning and memory. The way these neurons communicate to each other is by releasing those orange dots that you see there that are neurotransmitters. And these neurotransmitters are produced inside the neuron, packaged in these little vesicles, the little blue bulbs that you see there, and then released so they can communicate to the next neuron. When we look at this system, it's actually way more complex than the picture that I just showed you. It's like this constant trafficking of proteins and vesicles inside the cell. We see the vesicles, so the little blob is taken away from the membrane, brought inside, carrying proteins and other neurotransmitters inside to what we call early endosomes. Then it can be brought back to the surface through recycling when needed on the surface. It can be brought back to compartments, other compartments in the cell. It can be transferred for consequent release or for degradation. When we don't need a protein anymore, this is the system that takes away that protein that is not needed anymore. What's interesting is that all the proteins that I mentioned, so alpha-synuclein, LRRK2, BPS35, RB8, and others, all these proteins that can cause Parkinson if modified genetically, they're all working in this system together. So these are several types of Parkinson. They all present with the same symptoms. They're all caused by alteration in this same system. So this is very interesting, obviously. What's also interesting is that this system is degrading proteins, and one of the proteins this system is degrading is alpha-synuclein itself. So the same protein that we see aggregating in Parkinson's disease is degraded through this system when it's in excess. So altering this system can also cause all the typical pathological forms of Parkinson. So we have all this genetic information now. We can create mouse models carrying the same mutation, and obviously, as many mutations exist causing Parkinson's disease, there are many animal models that can be created. And we are currently working in our lab on the first 4 of this list, but there are many others available in research. Information that we got in the past in the literature from animal models, one mentioned by Silke earlier, the mouse that is carrying modification of alpha-synuclein. So this mouse is carrying more alpha-synuclein than it's supposed to carry. These mouse, early in the age, young mice, actually have the same increase in dopamine. And then later on, they lose that dopamine. So they're presenting the same characteristic that we see in humans. And so we can compare these 2 models. So now I'm going to move on to BPS35 because it's the main focus of my study. And BPS35, we know, is involved in that trafficking, in moving around those proteins and those vesicles in those neurons. And we know that the mutation, specifically D620N, is what's causing Parkinson's disease. So we created a mouse model that is carrying the same mutation in the mouse genome. So the mouse genome is very similar to ours, actually, and they carry the same mutation that we see in humans. We're going to call them BKI from now on to specify when I'm talking about these mice that carry the mutation. And so now we can look at their behavior, we can look at their brain activity, and we can see if we can replicate what we see in humans in this mouse. So we have several tests that we can use to test their motor activity, for example. Here I'm showing what's called rotor-rod test. You place this mouse on a rotating rod. The mouse is happy there to run, but eventually will get tired, will fall, will fail, so will hold on to the rod and rotate with it rather than running. So you can measure these events, how many times they happen, and if there is a motor deficit, then you'll see more of these failing events. Another way to look at their locomotion, you just place them in an empty cage and track them with a specific software, so you can look at their path, where they're moving, the distance they covered, and how much time they spend moving. And this is, again, is an indication of like proper motor activity. Another one is the cylinder test. You place the mouse in a cylinder. It will try to escape the cylinder, so it will perform rearings. It will also groom. Like, mice are very clean animals. They spend most of their time grooming and cleaning themselves. So those are also indication of like motor activity. This can be quite a fun test because some mice can be very, very active. So they actually jump out of the cylinder sometimes. But what we know is that when we look at young mice, so these are 3-month-old mice. Mice have live around 2 years usually, so they're very, very young. They don't show any motor deficit at this age. But what about their cognition? So we can put them in several types of mazes. Here I'm showing what's called radial arm maze. As the name says, is a maze with like different arms, 8 different arms. They radiate from the center. So the mouse is exploring this maze, starting from arm number 1, and we can put a reward in one of the arms. So in this case, in arm 8, we're placing sucrose solution, so pretty much sugar. They love sugar. When we do this in healthy mice, we see that in the graph on the top is showing that mice are spending up to 30% of the time, of the total time of the test, in the arm with the reward, where they're only spending about 10% in other arms. You place them there the next day, they behave the same. You place them the next day again, and they behave the same. Then we move the reward into another arm. So now the reward is in arm 4. They stop going to arm 8, they reduce the time they spend in arm 8, they increase the time that they spent in arm 4. They're recognizing that the reward is now in a different location, so they moved. They do the same for the 3 following days if you keep experimenting on them. When you remove the reward completely, they stop going to either of those arms. They explore the maze in general, but they don't spend much more time in one arm than another because there is no more reward there. When you do the same in the VKI mice, so the mice that are carrying the mutation, initially they do spend more time in the arm where the reward is, so arm 8 on the graph on the top. But then when you move the reward into another arm, it takes them longer to realize that the reward is in another arm. So they still go there eventually, but they don't spend as much time in their arm because they're still thinking that the reward is in the arm 8, where it was initially. So they're a little bit slower in this cognition task. Another test that we can use is the elevated plus maze, and again, the name says what the test is an elevated maze. It has a shape of a plus. It has 2 arms that are covered and protected, and 2 arms that are exposed. So mice are prey, so they're afraid of a predator coming from above, usually a bird. So they don't explore much the open. But they're also curious. So eventually, after 5 minutes of exploration, they will try to see what's happening out there. If they're particularly anxious or particularly scared of the situation, they will spend less time in the open and more time in the enclosed arms. And this is what happens in these mice. Overall, I'm showing here in black the mice carrying the mutation, and in white, healthy mice. And they explore the maze less. So the number of entries in each arm is lower. And also, they spend less time in the open. So the actual time that they're spending in the open is lower. So what we're showing is that these mice at this age, 3 months of age, don't have any motor deficit, but they already show cognitive alteration, which is often what we see in people with Parkinson. So what about the neuronal activity? I talk about dopaminergic neurons. I talk about this release of neurotransmitters. So dopamine is produced in these neurons out of like specific molecules. So there is a specific chemical process that produces dopamine in the neuron. Then once produced, dopamine, those blue dots, is immediately packaged in these vesicles to protect it from degrading. And then through these vesicles, dopamine can be released outside of the neuron to communicate to the next neuron. And then it can be either taken back. So that protein that you see there, DAT, is actually a protein that is taking back the extra dopamine, so we can reuse it. It's a very efficient system. Or dopamine can connect to the next neuron and have the function that it's supposed to. So we can use these proteins, dopamine, DAT, and other dopamines that are either producing dopamine or transporting dopamine to identify these neurons that are dopaminergic neurons, and we can use them as a quantitative evaluation of like the structural integrity of these neurons. So if you lose these proteins, you're losing those neurons. So we can take the mouse brain. We can cut the brain, get specific sections, look at the area of interest where those neurons are present. And we can give pretty colors to the specific proteins and quantify them. And we know that structurally, at this age, 3 months of age, these mice are fine. They're not losing any dopaminergic neurons. Their system is still intact. But what about their function? We can stimulate electrically this area and then record the response. And this is what it looks like. You send a stimulus, and you get a response because the neurotransmitter is released. And the bigger is the stimulus, the bigger is the response that we record. So what happens in healthy mice, you're sending bigger stimulus, it goes from 100 to 700, and the response is bigger. What happens in the mice with the mutation, they're actually releasing more dopamine. So again, at this young age, they don't show motor deficit, but they have increased dopamine and cognitive alteration, while the system is intact. So there's no losing those neurons yet. Another way to look at their dopamine levels is you can actually do it in vivo, so when the mouse is still alive. So surgically, you can implant a tiny probe in the brain of the mouse and use this probe to collect brain samples while the mouse is behaving normally, is doing its own thing. And what we're doing is we're taking all those dots, all those neurotransmitters, and we can quantify them. We can actually tell how much of the neurotransmitters is in the brain. What we're getting mostly is actually the next step of dopamine. Dopamine, I told you, degrades very quickly. So once it's released, this is right away converted into other molecules. And we can quantify those, and they're increased in these mice carrying the mutation. So again, this is another proof that dopamine is increased at this young age. And this is exactly what we were seeing in human carrying a different mutation on a different protein, but presenting the same disease. So now we can put in this graph that I showed you earlier, where we have the human progression of Parkinson's disease and other different mouse. We can put our mouse right there. We have also this increased dopamine in early stages with cognitive alteration. Now, obviously, we're looking what's happening when the mouse ages, what's happening if we lose dopamine later on. We're still studying old mice, but we know that they do lose weight. So they don't weigh as much as healthy mice. So something metabolically is changing in these mice over time. What else we can do? We can look at the cellular level now. So we have the same mouse. We can take neurons from these mice and grow them in a dish. So we can look at the single neuron, what's happening in the neuron when this mutation is present. We can look at the structure of these neurons, and structurally they're normal. So there's no change in the integrity of these neurons. They look as complex as any other neuron from healthy mice. We can look at the S system, the trafficking system that I showed you. We can actually follow proteins and vesicles moving around in the neuron. We can track them and see if that is altered. We can look at their neurotransmitter release, because even in a dish, they will still be active and release neurotransmitter constantly. So we can quantify that. We can look at dopamine and metabolites of dopamine, and we can modify this very easily. We can increase the amount of dopamine that they release, decrease the amount of dopamine, bring it back to normal. So these are systems that we can modify very easily. We can look at the single neuron activity. So how much each neuron, not the entire dish, but each single neuron, how much is releasing and how often. So this is what it looks like when they release another neurotransmitter, which is glutamate. You have each one of the spikes is a release of neurotransmitters, and these happen constantly and a specific frequency. These mice, these neurons from mice carrying the mutation, they are actually releasing more neurotransmitters. You see that there are bigger responses and more frequently. And this is, again, is a model that we can easily alter. More than a mouse, more than a human, we can apply drugs here and see if we can reverse that activity. So this is why the importance of this science is, because we can use these system to see alterations that are early in the disease that we can use to identify specific populations, so we can identify people that will develop Parkinson before they actually start to lose those neurons. And we can use this system to develop therapies. This is something that we can easily modify, and so we can understand what's the mechanism, and now we can change it before it's too late. And we can bring this information back to clinical, so we can help people with Parkinson's disease. So I want to thank my group, my lab, my supervisor, Matthew Farrer and Austin Milnerwood, and all the people that helped me in this research and this work, and you for inviting me today.